in frequency domain. So given a signal in time domain and sampled it through an analog to digital converter, how we can analyze it through some discrete techniques or through some digital computers, we will discuss these concepts in detail in this section of our course. And most importantly, we will also discuss several benefits of representing signal in frequency domain, the benefits which we cannot have by representing our signal in time domain. So let's start with some basic terminologies which you should know before proceeding further. And I hope all of you are already familiar with these basic concepts. But just for the sake of revision, some key definitions and some important points are presented in this lecture. So let's start with what is analysis. Analysis is the process or procedure of splitting or breaking some complicated looking phenomena or some complex substance into its simpler elements or components in order to gain a better understanding of it. This is the definition of analysis. Now I would like to discuss a very good example of analysis which we studied in our chemistry course at intermediate. Suppose a sample of some unknown compound is given to us and we are asked to perform its analysis. So what does it mean? Compound analysis means we need to find two things. Firstly, what particular elements are present in a given compound, and secondly, how much of each element is present in a given compound. So the first analysis, which deals with determining the type of element present in a compound, is called qualitative analysis, while the second analysis, which deals with determining the quantity of each element present in a compound, is called quantitative analysis. And the idea of signal analysis is very similar to compound analysis. That's why I discuss this example here. Now, what could be the possible objectives to perform analysis? It might include to inspect or to investigate the nature of something, to perform detailed examination of the structure of something, to observe the characteristics or essential features of something, or simply to study something. So this is the basic idea of analysis procedure. Another important concept involved in this discussion is synthesis. What is synthesis? Synthesis is a process which takes two or more simpler or smaller elements or components and add them together in order to produce some complicated looking quantity or some complex quantity. Why the resultant quantity is considered as the complicated one? Because it's nothing but simply a composition of some simpler modules or some simpler basic building blocks. In order to explain the idea of synthesis process, here I would like to present two examples in order to gain a better understanding of it. The first example which I have selected is from chemistry. If you can recall, we studied synthesis reactions in chemistry. What are synthesis reactions? Synthesis reactions are the combination reactions where two or more reactants combine together in order to produce a single complex substance. So synthesis reactions are nothing but simply they take some simpler quantities, mix them together in order to produce the resultant complex quantity. Another example of synthesis process, which is a very good example, is color synthesis. Color synthesis is a method of creating colors by combining by combining various proportions of some primary colors, and the most commonly used primary colors are red, green, and blue. So we can create any desired color by mixing these primary colors in various proportions. So if we will combine red and green color with an equal intensity by keeping blue color's intensity 0, the resultant color will be yellow. Similarly, if we will mix red and blue color in an equal proportion by keeping green color's contribution 0, the resultant color will be magenta. And if we will mix red, green and blue color in an equal proportion, the resultant color will be the gray color. So the idea is very simple. We can have as many as possible colors, or in fact we can have various shades of a single color by changing the strength of each primary color. So by changing the contribution of red, green and blue color, we can create a variety of colors. So the basic purpose or objective of synthesis process is to create something, to produce something, to develop something, or to make something. So the idea of synthesis process is totally opposite to analysis, which we studied earlier. Here, a very good example of both analysis and synthesis is presented, and it is a very famous experiment which was conducted by Newton in 1672. He demonstrated that white light or sunlight can be broken down into its 7 component colors. So, if we want to analyze the white light, we need a certain tool, which is a prism in this case. Prism is just a tool which enables us to observe the constituent components of this white light. So that if you will pass a white light through a prism, then we can observe its component colors, where each color will be having its own unique properties. So the dispersion or splitting of white light into its constituent components or into its ingredients is called an analysis procedure. Conversely, if we will combine or add the 7 component colors, the resultant quantity or the final quantity will be again the white light. So the procedure or the process of adding or mixing the 7 component colors in order to get the final white light is called synthesis procedure. And in this example, the white light is considered as the complicated looking phenomena. Why it's considered as the complicated? Because it's composed of some simple colors or some basic colors. So this phenomena can be simplified in terms of its ingredients, where each ingredient is offering its own unique properties. Similarly, these component colors will be considered as the basic quantities or the simpler quantities, because each color is different from the other by its own properties. So hopefully you get a better idea of both analysis and synthesis by discussing this example.